Okay, maybe we can get started. Today, Auke is gonna be give us a presentation about the MIAPPE. Before we do that, Peter, do we have anything that we need to discuss about that poll that we want to send it out?
I have made some updates that we could review. Yeah, I don't know if we want to do that now or later, but yeah, I did take some feedback from the last discussion I was able to attend and
Okay.
made some updates. So yeah, up to you when you want to do that.
Okay. So Auke, how long do you think the presentation is going to take? And it depends on if it's not too long, maybe we can just put you some of the remaining hours to talk about the survey.
Okay.
Well, I have 5 minutes, and then I've asked Rick to share some more about MIAPPE because he has more expert info, and then I think we can, yeah, make it as long as we want
Okay.
because it's also a bit discussion style. But yeah, let's see how far we need. But
Okay.
I'll just give a brief introduction and then pass it over to Rick. And I don't know, Rick, you're on mute.
Yeah, I can whatever you want. Actually, I want to launch a discussion on MIAPPE implementations in a applied phenotyping center. And if you need me for an hour, then I can do it. Actually, if you say better summarize it in 10 minutes to launch a follow-up discussion, then I'm fine too. So whatever you want.
Let's do it this way because I don't think we already talked about this survey for quite extensively in our previous
Yeah.
meetings. Now, I don't think we're going to need a lot of time. Mainly review whatever the changes has been made by Pete, and if everybody's okay with it, we can send it to Narita to send it out. So please take as much time as needed because this is going to be the main topic of today's meeting. So whenever you're ready, Auke and Rick, please
All right.
take it away.
Okay, let me just share my screen. Um, more. Where is the sharing button?
Good morning, Anna.
Good morning.
Okay, share. So, do you see my screen?
Yeah,
Yes.
perfect.
Okay. Yeah, so I'll just share very briefly about CropXR, which is the program we run and where we are implementing MIAPPE. And then Rick is also connected with MPEC to CropXR, but he also has his own institute. So, but he will explain. Um, but CropXR in a nutshell is a public-private research institute dedicated at innovation for more sustainable and climate-adapted agriculture worldwide. So our focus is really on crop resilience, and we have a few varieties. So we focus on brassicas, chrysanthemum, lettuce, onion, potato, and tomato. But we also look at the general topic of resilience, and we use our Arabidopsis as a model plant for a lot of fundamental research. So we have some funders, some partners. We're funded by the Dutch government with almost 100 million euro to spend. So we can, yeah, make some impact, hopefully. And it's not just universities who are joining, but also a lot of breeding and technology companies. So we're really working together with a lot of partners. So standards are super important. So we all speak the same language. So just our mission in 2033, we want to have developed resilient crops through data-driven design on the ground. So that's an ambitious mission, I think. But well, it should be feasible with all the breeding companies also on board. So just what we do. So we have this score, which is called PlantXR. That's where we do the fundamental research. And then on top of that, we have a few programs, education, but also in data. I'm responsible for the whole data infrastructure. And then we develop this fundamental research. And then in a few years' time, then the companies, and that's outside of the blue box, they should develop products. So all things that we do is pre-competitive. So that's why the companies are working together with us and together between the companies because it's all pre-competitive research that we do. But eventually, of course, there should be products resulting from the research that we do. So then on metadata, just a few slides, we have some groups. We call them SAME groups, standard and metadata experts. And we have two groups, one on phenotyping and two on RNA-seq. And on phenotyping, well, we base on ISA and MIAPPE standards. And RNA-seq, we have a whole different view. Looking at NCBI, ENA, and all the standards that are there. And then, so we try to detail the metadata workflow, and here's where this MIAPPE comes into place. So we really want people to register an experiment before they run the experiment and download a pre-filled Excel file based on MIAPPE before they run the experiment. I won't go into all these steps, but the basic thing is, okay, just think about your metadata before you do things. And also we want to build in some things that you won't get your data after you filled in your metadata. But we're still in the process of thinking about how can we do that. To just ensure that people fill in the metadata because nobody likes it, but it's beneficial to everyone if you fill in the metadata. These are the metadata fields that we have right now. So it's for sequencing on the right, in the middle is phenotyping, and on the left are some generic fields based on ISA that are for our program. So the focus of today is on the middle, but Rick will go into that more. And then I have one final slide before I give it over to Rick. And that's what our vision of the future is in the next step, to support researchers to fill in the metadata. And that's what we call a meta buddy that we're developing right now. And it's a virtual assistant for collecting metadata. So it's a sort of a chatbot you could chat to, and then it fills in the metadata for you. So that's something that we want to develop together with Wageningen and a lot of other initiatives to really help researchers do this tedious work of filling in the metadata. So just a teaser on our program and how we use MIAPPE. So we're really in the beginning, and Rick has a lot more experience. So we're working together with his group of MPEC to implement this.
Yeah. Thank you.
All right, Rick.
Yeah.
If there are no
You
questions,
can stop sharing.
I'll stop sharing.
A quick question to the ones now in the room. Are you implementing MIAPPE metadata practices for phenotyping tools? No?
Is this a question to the
audience? Yeah,
audience?
maybe to the recording, but yeah.
I mean, at least for me, I don't know the detail about this MIAPPE. I think I'd like to learn what it is.
Okay.
And
No,
we
just
have
to
our own platform to manage all this phenotyping data. So we are interested in implementing it in our system. I think others might have a different answers.
Yeah, and
No,
sort of, I'll say at the other end of the scale, I run the BRAPPE project, which is fully MIAPPE compliant. So there's
cool. Okay.
full range of audience here.
Yeah, okay. So what I will do, I will drop a lot of ideas. I will go a bit fast. and hopefully I'll create some hooks for a further discussion, because there's a lot happening on the level of metadata implementations with international partners, really from Japan to the US and back again, with Europe in between. So, okay, let's start sharing. Of course, you know this. What I'll do, I think I need to take you with me with some background information on why we are implementing metadata. But we do it because we generate an enormous amount of data on fields like this, but also in greenhouses and growth rooms. And then projects like this want to study lettuce. They want to figure out why a certain variety or a certain genotype is growing different because of treatments, because of weather patterns, because of soil characteristics, or maybe because of genetic variation. And if you capture this kind of data over the years, then the metadata becomes essential to make sense of the raw data, which is big pictures. So if you have a picture of this single lettuce plant, you need to know where it is, when was it recorded, with which camera, maybe even who was driving the vehicle, which camera, which type of camera, which brand, and all that metadata needs to be captured in a very accessible way. So that's a big challenge. And I've got many different types of sensors, and I would like to close with a very applied example with actually these two technologies to merge the data from both viewing angles. So I'm Rick. I work in Wageningen. I have a background in AI, and I'm the Chief Technology Officer. I think that's the main role in this talk, at least for the Phenotyping Center here on our campus. And hopefully I'm going to inspire you a bit or launch some ideas. Maybe you can learn me something in the upcoming near future. But it's all about this, of course, this equation. We have many genotypes. We would like to screen them in all kinds of environmental settings, conditions, stress tests, and then you get some kind of phenotype. We use this equation to get money from our government to say if we would like to study many genotypes, we need money to screen a lot of phenotypes. So we got money to close basically this gap. So there we have already in this university hundreds of thousands of researchers that could benefit from large-scale screening of plants. The experiments should be bigger. They should screen more genotypes. They simply should discover robust varieties faster. So speed up the whole breeding discovery process. If we would give this majority that is not benefiting from the technology access to very expensive tools, their research output will increase. And we can close the gap between high-tech tools and the breeders, the growers, plant physiologists, etc. So we got the money, also like CropXR, we already received it 5 years ago. We started building a large-scale research infrastructure for phenotyping, and I'll show some pictures. And any day you can visit us, we are open. We get many visitors from all around the world to team up with us and also develop, especially software, data, tools together with us. We post a lot on LinkedIn. If you want to follow us, just do. Now, MPEG is all about phenotyping in several types of conditions. So below ground, high controlled ecotrons till completely uncontrolled field conditions and everything in between. Plant microbe interaction, high throughput growth rooms, greenhouses. Many users, many providers. So they sold equipment to us basically, and many international partners. Because of time, I'll just keep on talking fast and flashing some slides until we reach the fair stuff. So it's just a few impressions. This is growth rooms. Lots of plants are imaged every day in different settings for all kinds of reasons. And then we get time series data of all these plants growing, captured with many different types of cameras. And then we see differences. Then we would like to explain these differences. The same goes for the greenhouse, just an example of many tomatoes in a conveyor system where we image them every day with all kinds of cameras, generating, of course, terabytes of data. These plants can travel, so it allows us to make beautiful, also full 3D reconstructed shape analysis tricks. Just to illustrate it, plants grow over time, and especially time point analysis is interesting because you would like to introduce a stress and then figure out when the plant responds to that stress. Yeah, so coming basically to the essence of today, we generate a lot of data, pictures, sensor data. We bring it all together. So this is a scheme from a very applied point of view. So millions of pictures. Let's put them in subfolders so you can find them, label them, create some first analysis steps, visualize those results, because we found that most of our users, as well industry, international as Dutch, find this complicated. So they find it complicated to receive millions of pictures because they want to figure out which genotype perform best. So there was quite a gap between users. So some users find it very pleasant to receive millions of pictures. Most find it very disturbing, and they just want to have graphs. And we serve both ends. So just to illustrate it, pictures are created, data files are created locally on a PC. We are uploading it to a cloud provider like Azure or Azure. There is some post-processing happening. People can log into this process, check their data live as it is generated. After a while, we put it in archive, but otherwise it gets too expensive. And then people can download it through iRods, iBridges tools to allow them that allow us actually to store that data for 10 years. Otherwise, in Azure, it would be costing a fortune. And then we also provide additional data, insights, which is, for instance, this Python notebook to show how certain graphs were generated, and users can play around with these graphs and generate their own version for their own specific research question. But this is all pretty data oriented. And now I would like to zoom into the metadata challenges, because we all said, okay, in our data management plan, we're going to deliver fair data. So we need to make it findable, accessible, interoperable, and reusable. I think this is extremely complicated to do this completely 100% robust. Words that you use, but that's where ontologies come in. But to implement that, you need to have something very stable, and you need to be able to search through all that metadata to be able to find that we done 10 years ago some beautiful experiment that somebody actually requested today, and we say you don't need to do that experiment anymore. We already did it. Here you have the data, but then you need to figure out in all these experiments if there is a real match with the future experiment. So some tools that we use, MIAPPE. I think MIAPPE is not a standard, to be honest, although people refer to it in a way, but it's not as defined, as specific that it can really be implemented. Let's see if somebody triggers on this statement. And we use the ISA model to really structure our data also in folders and subfolders. And then you have certain words that are used in the ISA model, like investigations, studies, and essays. And we are also, to be honest, still struggling a bit how to match all the concepts that we have on these words. And that's the main focus of today. So MIAPPE, is it really an international standard? It is commonly known. It is accepted. People refer to it. We have been contributing to it. And it helps. An enormous step in defining what is metadata, what needs to be minimally collected, so minimal information about a plant phenotyping experiment. But it's not to me, it's not a completely implemented standard, but we are on the way. So I would say MIAPPE are very useful guidelines. Where we are now struggling is actually the combination with ISA and MIAPPE. And I would like to show just in a few slides that why we do it. Jumping back to a trial on the field. So if you have one project, title is Tada, has multiple experiments in different countries with 5 times flights of a drone. So here you can see flight 1, flight 2. You can see actually the potato plants slowly growing. And then we need to figure out what are the level of detail. So what is actually the assay here? So what is really the measurement? We said the assay is actually the plot where we are measuring which what a single genotype is doing on this specific zone in the field. And how can we match that with the growth rooms? So what are then the assays, the measurements? How did we take our sample? What are the observation units? What is the study? Called the investigation, the project. And we found that in Fair Data Station, there was an implementation of MIAPPE. So you can actually select it. You can generate an Excel file, which CropXR Auke also referred to. And you can fill in this Excel file. And that feels already quite complete. But if you then really analyze the different fields reported in MIAPPE in this Excel file and what we actually need, then we can find all kinds of nasty differences, which might be essential. I'm not going to go into details because then it will take me really an hour. But this is really ongoing work, and we would like to align on all these specific minor, maybe very essential differences. On the level of an investigation, on the level of a study, there are small differences. If we come to an agreement how to implement it, we can generate data files from any field, any greenhouse, any growth room in the world, exchange it, and the metadata files are then also identical. And we think some of the elements are missing because we cannot store it. Well, we think it's essential to store it. This will trigger a long discussion, but we need to go through this tough phase. And we're doing it together with many international partners, also from Australia, from the UK, from France. So we're not doing it alone. If you want to join, from Germany too, US too. All triggered by the International Plant Phenotyping Symposium we had in Nebraska 1 month ago. So what is a sample and what is an assay? So the assay is the measurements. So we found that there are some differences that are really essential and need to be added. Otherwise, you cannot link back to certain elements. Okay, and then of course there are ontologies underneath. We also found that not everything is completely alive anymore. Some things are not curated online anymore, so that's a bit disturbing. Okay, I would like to show you one example, also why we do it, hopefully to also inspire you a bit. So if you have curated data, so you have many experiments, one experiment has a specific pot, we got hundreds of pots, it has a certain code that makes it unique, and there's a certain date-time combination. When we did imaging with a certain sensor, we had generated some point clouds, we derived some data. So people can go through this folder and find their data, but also machines can read through these folders and retrieve certain data. Or you can write Python code that says, give me only point clouds with this and this specific genotype. If you do
Rick,
yeah. Cool.
Just a question on the previous slide,
yeah.
The folder. So MIAPPE is based on ISA, right? But this folder structure is not a MIAPPE folder structure.
No, you're correct.
No, so it's something you then developed yourself based on ISA.
Yeah, exactly. Or... No, but that's the whole thing. This is loosely inspired by ISA. Yeah. Okay. So here there's not a standard. This is our way of implementing data in a way that we can find it, and we can write code to retrieve it. Yeah, good comment. Very good comment. And, these are the nasty small differences that might bother you if you get this data set. Okay, and to illustrate why we do it, so we have drones flying over a field. We define plots. Plots are the measurements where we actually extract information from. We have another device that drives over the field to collect the data. Just to illustrate it, it's a massive imaging cabinet. So it drives over this field, captures image data with a hyperspectral camera, with a 3D camera, and a color camera. And he's just piling up data. And we bought this machine. And at the moment we bought the machine, we asked the provider to make all the metadata that he could generate automatically available through a JSON file. And that helped us enormously because we have plots, right? We have driving directions. We know where this machine was driving. This is an orthomosaic of a potato field. And the driving lines of the vehicle were actually plotted on the drone image. And this is hopefully handy. We said, if you would like to capture all the metadata, it is annoying, right? As Auke already mentioned, for the research to fill it all in. It's time consuming. They might not even have it available. So why not try to fill it in as much as possible with all the machines around us? So this driving machine is generating this JSON file, and he's just spitting any value that he can find into this file, which is some environmental conditions, information about the GPS coordinates of the plot. He knows that actually, but also when he was driving in this field, but also how he entered that plot. So the planned entry coordinate, which LiDAR sensor was used, which type, which product number, which serial number, and how was it mounted. It could all matter. So, and we can generate it. So the user doesn't need to be bothered with this. But metadata is essential. If you, for instance, would like to fuse the drone data with the trade secret data, and we are still puzzled by small differences implemented by our providers. So we have a test case on a concrete lane in between of our test fields on campus here. We placed some calibration objects on purpose at a specific point. We created plot files on specific GPS coordinates, and then we started flying and driving to capture data, and then you get these kinds of data files. A drone captured an RGB picture, trade secret even the driving direction was reversed, so it was flipped, for instance. All these nasty small things matter. And to me, that's also metadata. The metadata of the driving direction was added because we once in a while drive from this angle into the field, and then we turn and we drive backwards to the other end of the field. Okay, in a nutshell, but I would like to hear what you think. We need to explore this together. So it's all about information extraction from the raw data, which traits, but also which metadata is needed. And we have the philosophy that if we do it together, we can develop more tools. So if one smart guy is developing some very smart AI implementation to extract some information from some point cloud, he has his tool developed, he shares the code with the next experiment, then we simply get higher quality output every year. People add to the source code that is then collected by our phenotyping center, and we provide access to it. And the team is currently very focused on data, AI, and hopefully this triggered you. So how long it took me? A bit longer than 15 minutes, maybe. Sorry for that. Yeah. Done.
Thank you very much for the very high-level summary. I'd like to open up the stage for any question that you might have from the audience. Anna, please go ahead.
Hello. And thank you for the presentation, both of the presentations. They are fascinating. I have a question about, you said that you're combining ISA metadata model and MIAPPE metadata model, and also that you were kind of like, so are the vocabularies provided by those standards, or are you bringing in vocabulary from the ontologies? And were you able, or how easy was it to merge those two approaches, and how did you select the controlled vocabulary to use to kind of like for the metadata fields or for the metadata names? Might you be able to tell us a little bit? Because in my opinion, that can be quite tricky. So, and you said that you found that some of the metadata are missing from both of these standards. And so how are you going about, so, whichever question is easier to answer, like, how are you going about like adding those? Because this can also be very tricky, kind of like, how do you extend those standards and how do you work on that?
Yeah, that's I think that's the essential question. We didn't implement, or we implement a lot. We didn't define any of these metadata fields. We just did a thorough analysis of what MIAPPE was describing, what we found in ISA, and how they were implemented by some partners, and what the differences were. But I think there is a lot of knowledge already out there with people doing this, and we can just embrace what's there and come to a common understanding. I'm quite sure that Nathan or Peter has a strong opinion on this too, but to give you the floor. Yeah, so we are just exploring what is out there. We are trying not to reinvent any wheel. I think there are some smart guys implementing it, and we are just reaching out to those that developed some of these codes or documents and say, hey, I think there is something missing in your definition. That's what we do. Yeah.
Okay.
Maybe on your question on ontologies. So in CropXR, we just said, okay, let's use the Plantiome ontology for all terms around plants, because there's several ontologies. So we just said, okay, let's use that one. And then there's the big issue of that there's gaps in the metadata fields. So yeah, we added a few fields, but also because we're also looking at sequencing, so we also have a lot of extra fields there. But we really try not to change the existing fields, just add stuff.
Thank you.
Yeah.
That's helpful. Thank you very much.
And yeah, Rick, you called us out here. I'll jump in with my thoughts. So from a BREPI perspective, I get to from a BREPI perspective in terms of terminology, I get to be a little bit more picky because BREPI is designed for computer readability, and computers don't care as long as you use the same word every time. They don't care what that word means.
Yeah.
And so, I've the number of times that I've had the conversation with people who, MIAPPE has investigation and study versus BREPI, which is trial and study. You know, the number of times I've had conversations where people want to change the BREPI spec to be investigation and study or whatever. There's many different words for that. And I say, the computer doesn't care. As long as we can clearly define, like, if you're talking in BREPI, this is what a trial is. If you're talking in MIAPPE, this is what an investigation is. Clearly define those so that we as humans can understand them. The computers don't care what it's called and when they're exchanging data. So I'm a little bit lucky in that sense that I get to make that argument for BREPI, and pretty quickly shuts down any sort of that sort of naming issue. In terms of changing the spec, I 100% agree with both Rick and Auke in that yeah, I do our best not to change existing things, but add new things. And sort of to add on to that, these standards are living documents. And they are all versioned. So you have different versions of the standards. Um, and as the community progresses and as we that will continue to be the case. We will never come to a point where MIAPPE has every single metadata field that is perfect in every way, because as the science progresses, uh, drone imaging was not a thing, 5, 10 years ago, and now it is a primary way of collecting phenotypes, or it's becoming a primary way of collecting phenotypes. So that's going to change the way we're looking at the data standards. And so pieces need to continuously be added to accommodate new science and new research and things like that. Balancing that with the stability of being a standard and having everyone being able to rely on something being standard and unchanging. So there's a balancing act there in terms of um, adding new things as they become relevant versus keeping something dependable that people can rely on.
Yeah, Peter, can I ask you something now? Because there's something very specific that we are now working on is if the drones, you mentioned the drones fly over a field, we set the plots are the measurements, right? So we extract a sample from our whole field, from the study. We do a measurement on the plot. The plot has a certain ground coverage and a certain amount of plants or a certain greenness value. Okay, but now drones get a higher resolution, and within this measurement, we can do another measurement because we can zoom into an individual plant and say, hey, we got 20 replicates within my measurement, and it doesn't fit with the data model actually. And the next phase already comes in that people say, I want to count the leaves of the individual plants, and actually I can see traits on the individual leaves of the individual plants. So it's like this Russian babushka doll, right? It always gets more detailed, and not only on the field. It's for every phenotyping sensor is diving deeper into an individual measurement. How do you capture that, and how do you actually need to be something like a recurrent, reiterative process that you can loop into itself? How do you deal with it?
Yeah, it's certainly a struggle we've been facing in the RAPI community. The way we're dealing with it right now is linked observation units, and so you will have an observation unit that represents the plot and has all the metadata surrounding the plot and some observations connected to that. And then you'll have another observation unit entity that represents a specific plant within that plot. And there's a connection between those two observation units saying this
Okay.
this observation unit is a plot, this observation unit is a plant that belongs to that plot. And dive, and then you can, with that structure, you can dive deeper if you need
Oh,
to.
yeah. Oh, cool. Okay, then we align. But that's what's actually happening here. The observation unit can be a plant and a plot and both. And then we can still create indeed this loop where you have data from a plot, and a level deeper you got the same plot which then generates data from a plant. So okay.
And there've been a few cases where actually we've had like you even go up levels. So say we have this observation unit that represents a field,
Yeah,
and
that's another one for temperature.
temperature or connect the orthomosaic image, anything you're collecting off the orthomosaic, that sort of thing, to the field level observation unit, and then dive down from there. So yeah, it's not perfect yet. There's some it's a little bit difficult to navigate if you have a really complex study with, you know, 10,000 observation unit records that are all different types of things.
Yeah, I know. Yeah, we got the metadata files with 80,000 rows or
Yeah.
more, and then people want to open it in Excel. It doesn't work.
Yeah.
Is
Yeah.
there any attention that's being given to creating a language that pays attention to our language? I mean, these are things that are commonly done in computer science constructs. I mean, for those of us that program hierarchical inheritance, mildly complicated. The mathematicians have done this when they talk about fiber bundles and mapping and parameterizing different spaces. I just feel as though there's, at this point, potentially a lot of value in cognitive abstraction. In other words, not dealing with the specifics of which fields are the right fields, but rather the structure of thought and language required to do recursive embeddings. And to be able to communicate with the biologists and the breeders at that level might provide for a more fluid conversation when it comes to talking about the difficulties, because the biologists inherently know this. Like, we know exactly how complicated our studies are. And the computer scientists and the mathematicians and the engineers natively have this as well, but it's housed in a different part of their language set. And I just feel like kind of marrying together these two abstractions, so that we can talk about a plant is both a plant and a container which holds cells and a thing which we're going to embed leaf measurements to, and that we can handle these Russian dolls being abstracted out. You know, clearly I don't have it worked out. I just continue to find that language is both subtle and a horrifically powerful ally.
Yeah, I agree. Even if you start talking about specific plant parts, then they have names. But the data scientist and me, including, do not know those names. And it's very fuzzy to talk about very specific measurements.
Well, in the part which can be horrifically difficult is that at times the names of the domain on the tissue is functionally named, and how to call it the name won't happen until you phenotype it and find the measurement by which you then can label the domain on the plant. And so the name is a functional name, not necessarily an anatomical name, right? So, you know, there's all of these moving pieces, which makes it very hard.
Yeah, beautiful examples. We have a digital biomass, which is nothing, has nothing to do with grams or actual biomass. It's just an estimate. How did you make this estimate? Yeah, cool. Okay, I think this already helps. Glad that I get something back.
I was told that the limnologists have a paper out. I have yet to track it down, about a concept called flickering. And flickering is the situation in a conversation where a person moves back and forth between the abstraction of the plant, a specific plant, and a model for the plant. And that as humans, we move smoothly between these stacks without even realizing it. But when you come to formalize the situation, the person whose job it is to formalize which level we're talking about, we run across these difficulties, right? And so they named this flickering, where you're moving back and forth in a cognitive way without being aware of it necessarily, right? Between the abstract levels of abstraction.
So this
Thanks.
MIAPPE is a standard, or more like a spec, where you're saying that this is how you're supposed to your metadata, but actual implementation of MIAPPE is up to the scientists, whatever tool or in a software packages they can use it to generate this in a metadata, and that this metadata is available somewhere where people can just search through it and to find the data set they need, then just access it in whatever way that this data provider is, make this data set available.
Mhm.
So in that sense, I think that this is more question to Peter and Rick. So I think BRAPI is in the specification, but the way how you implement it is, kind of up to the user. So as long as they follow this in a standard, this data set is going to be fair. Is that kind of correct statement?
Yeah.
Okay.
So I have uh I've heard MIAPPE described um as a checklist rather than a standard. And maybe that's, it's a very advanced checklist, but it's it's sort of the agreement between groups of researchers saying these are the things that we all agree that you need to describe, to minimally describe a phenotyping experiment.
Yeah. Yeah.
And we don't, MIAPPE doesn't care how that data is stored, how that data is accessed, where it's located, whatever, but as long as you can go through um, and sort of check everything off the MIAPPE checklist and say, yes, I have this, it's stored over here. Yes, I have this, it's stored over there. Then that is MIAPPE compliant. I'm using air quotes. My webcam's not on.
Yeah.
Um, Things like ISA and BRAPI are a little bit more detailed in that they have a formal structure that should be followed, and the in terms of ISA and ISA tab, there's the folder and file structure that is well defined. And it says, okay, we're following the MIAPPE checklist. If you do all these things that ISA defines, that also checks everything off the MIAPPE checklist. Similarly with BRAPI, you set up a RESTful API service with these JSON data structures. If you do all that correctly, then you're checking everything off the MIAPPE checklist. Does that answer your question?
Yeah, so I mean the reason why I'm asking is because Peter, you've seen our platform where we have many all those different data sets. So we like to for example all these different data set, for example, BRAPI compatible, also, might be compatible as well. But since, Peter and you guys always spend a lot of time to, make this, standard available, I don't want to reinvent the wheel. And if you can just, work with you guys to make all these data set to be might be compatible, BRAPI compatible, so that others can discover these data set more easily. So I'm trying to see how that can happen where the way how I'm seeing it in Peter, I think I'm a little bit more familiar than Brapi, so that all those information about field layout experiment, all those kind of being stored in the Brapi database, and this Brapi is also might be compatible, and link this Brapi database with our data to science platform, so that we don't just implement everything you already did, but rely on this Brapi database, so that we can say that data to science is also Brapi and the MIAPPE compatible. Is that something that we can achieve, do you think?
Yeah, I think so.
Okay.
And and there are also tools out there for going in between BRAPI and ISA as well. So,
Yeah.
if you need to connect to the ISA standard, there are tools that exist to do that as well. So, yeah, it's and I think you're thinking about it the right way,
Mhm.
of sort of just the exercise of uh going through the data that you have in whatever proprietary format you've got it in, and doing the exercise of trying to implement the BRAPI RESTful API, or the ISA file generator, or whatever. Um, Going through that process, that exercise of trying to map whatever data you've got into one of these standards, if you can do it successfully, then you've got the standard model with all your data that can be uh, shared with other groups or whatever. And B, that also lets you go through the checklist and say, we don't have this piece of data that the standard says we should have. We need to resolve that. Or vice versa, we've got this extra piece of data that the standard doesn't have. We need to resolve that somehow.
Okay.
But yeah, just going through the exercise of sort of attempting to develop a mapping to the standard will open up all the little details and questions and everything that might come up.
Yeah.
Peter,
So,
you
yeah.
and I, sorry, like so you and I had started a conversation and we're working together to make this in a data science platform to be properly compatible. I think that could be some area. Yeah, your group and my group can work together to develop some collaboration so that.
Yeah. So we are now figuring out how to store all the data listening to the ISA nested folder structure, and then add all the MIAPPE relevant information at the right level in JSON files. Then you can place such a data set at some public repository linked to a publication. Everything I have, these are all my measurement files, all my metadata files. It's all text readable, so there is no overarching database needed to find some logical links to metadata fields. So if you have the files and the folders, the tar file, for instance, you have everything. I think that could work. I think if we agree how we structure that, then many people can benefit.
Question to you.
I think it's coming together.
The question to you is that a lot of example, I may be wrong because you just had a very high level overview, but a lot of example you present in your presentation is more focused on control environment than field setup, or at least I'm a drone guy, so most of my phenotyping is based on drone.
Oh yeah, no.
The
data set that we are struggling with is a drone data set.
Okay.
So we are doing both. Yeah.
So the way how it was presented in your presentation is more like, hey, we can actually see in a very high resolution images of in a single plant. Then you're gonna have certain kind of ID assigned to this in a plant, or the group of plant to be assigned as in a plot, and etc. So I'm not sure if I'm asking the right question, but how do you envision this in a MIAPPE can be implemented for the drone data set? Do you see that it's going to be almost same structure as kind of control environment phenotyping data, or?
Yeah, definitely. Yeah, that's what I hope. That there is it's not that the structures, the approach is identical, but the wordings are slightly different. Where we have in a controlled environment plots, and on the fields we have plots, for instance. Almost the same word, one letter difference. Got weather data, soil data on a whole field level, or maybe on a sub-zone field level. I think Peter, you addressed it too. There are a lot of phenotyping systems and software packages, and you can't change it because you're hooked to it. Somebody has a Lemmatech, another has a PSI system. So you can do whatever you want within your institute to collect your data and analyze it and organize it. But at the moment of publications, then you need to write some kind of extraction process to wrap it up, and then maybe only the part that you use for your publication, and put this in a public repository. That's my current approach, because most of these commercially available systems ignore the standards, so we cannot ask them to change. So we just need to deal with it and extract the information from the systems, and then publish it beautifully according some commonly agreed upon way. Yeah.
I'm still struggling if you really want to work with the data. So let's say you do MIAPPE perfectly. You have this checklist, but then the connection to the actual data, that's where I'm still struggling in how do you make that?
Oh no, that's. I'll show it to you for a second. It's very straightforward, I think.
Okay.
If you have a field, then you take a sample from that field, and then somewhere in your metadata file, so the JSON file, for instance, has listed all these things, plus the actual path, the local file path where your images are. Now this is the link to the actual data. You can say you got
But
a auto mosaic
I think that
located there.
this works for you, but what about the people outside of your institute? How they can access your data?
Because this is a local path to
Yeah.
a publicly available zip file.
Okay. This is
So it's
kind of
not down to a
background.
specific server, or so it's slash home experiment blah 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 essay this, but it's a local short path depending on where you downloaded this specific file.
Okay.
But even if you have that on a accessible place, it's just one link. So and it can contain hundreds of images. In drone
Yeah,
data, it's thousands of images. So
yeah.
how do
I'll
you
stop sharing. Yeah,
can you make sense of all those images? I mean, you have
yeah.
the metadata, but then you have a bulk of images. Yeah,
Yeah,
where
some
to
of
start?
our that's a some of our metadata files are bigger than the original measurement files. They're like millions of lines of where specific images are. So nobody's going to use it, but the machine can deal with it.
Ah, okay. So you do describe and make this connection to perhaps single
It must,
images.
yeah.
Okay.
Otherwise, there is no link to this image. You have
Yeah.
no idea what this image is doing. Yeah.
Uh,
This is
I,
a struggle because then these files get so huge that they are like text files of several hundreds of megabytes.
I think that that's exactly where I was mentioning earlier. Well, I like to work with the Peter's group too, because we have a online platform to manage those big UAV data set, which makes it available and accessible from anywhere. But we are storing this data set in a very structured database. But the way how you can discover this data set is not, I'm going to say, might be compatible, broadly compatible. So how can we link this in an online platform with a broadly MIAPPE, sender so that those community can access, this data set,
Yeah.
more easily. And I think that, Rick, you just mentioned that here's in a PyPath, but, there can be actual, URL to that, cloud optimized GeoTIFF, which is an automosaic that isn't accessible from, anywhere.
Yeah. Yeah, because not all data is public.
Yeah.
But on the other hand, you would like to know that there is some hidden data set somewhere that you can ask access to.
Yep.
So
Are you referring to the DS2?
Yeah, D2S.
You know?
Yeah, yeah.
Oh yeah, D2S. Yeah.
I'm not
Yeah.
sure, Rick, have you, I'm not, is this your first time joining this working group meeting? Or I don't remember.
Yeah, I sent Rick the link. It's his first time joining
Okay.
this meeting, but he doesn't have an account, I think.
I highly recommend you to watch that the presentation that I gave about a month or two ago. This is a
cool.
new online platform that we're developing, and if you're interested in, I think maybe we just we can work together at least to resolve that drone data set related data management and make it accessible.
Let's do it. Yeah, that's why I'm here basically. Yeah.
And I think I don't remember if it was in Rick's slide or Ox slide, but one of you mentioned like the necessity to produce visualizations and compressions and analysis, pre-analysis of the data. And so I think, I mean, what we've been talking about here, there are definitely two distinct audiences: those who are interested in the raw data, the zip file of 10,000 images plus all the metadata that goes along with it, and the audiences who are interested in looking at the visualizations and the summaries of things, you know, to help with decision support or whatever they're trying to pull out of that. And so there's definitely space and a need for both direct access to the raw data, um, you know, as a publication or just you know, as it's wherever it's being stored, and then tools and visualizations and support systems for uh, people who want to look at the data, but not all at once.
Yeah, I
Yeah,
think
I think I make a difference between like functional metadata and machine usable metadata. There are several levels of metadata. So indeed,
Yeah.
you don't like to scroll through many lines. It needs to be concise and short if you want to just check one experiment. And then, if you are really interested, then you would like to get maybe the full overview. Otherwise, it scares people away.
Yeah.
well, I think I like to echo, Peter's comment here is that a lot of it has, and I'm a civil engineer. I don't do any kind of agriculture science, directly, but based on my experience working with all those, breeders and agriculture scientists, every time I ask them, what do you need? Give me a spreadsheet, with all those numbers. But real value is that the actual original data set to derive that actual numbers is going to be very valuable asset here. So I think my kind of biggest kind of goal for developing this in a data science platform is that not only you can just access these numbers, but now you have access to the original raw data set to derive that numbers so that you can use different algorithms to come up with the new phenotypes and etc. So how can we just make that happen? I think that's the reason why I'm very interested in this data standards so that those scientists can will have flexibility to not only access those numbers but access to an original data set to generate that specific numbers.
Yeah, you're right. Yeah, and then we could go back to the files and add just a few columns to the measurements. Okay, we have some cool AI trick. Suddenly we can extract more information, and we'll just add it to a specific column, and we enrich the data file. Yeah.
Yeah, we can just keep talking about this, but definitely, I think Peter, I think well, after the break, maybe your group and my group can just have another meeting. I know already some of your members has been communication and our group to see how we can work together, but I'll be very happy to have another meeting with Rick, your group to see, and I can just give another demonstration of this data science platform to see if this might be kind of interesting
Yeah.
to your group to
Oh
integrate it
yes.
with MIAPPE.
Yeah.
Let's keep talking.
Yeah.
I know that we are already 6 minutes over the
Yeah, that's
ready
right.
time.
In my case, dinner is
Oh
already
yeah,
delayed now. In your case, your day is about to start.
yeah.
Yeah.
Any kind of pressing comment or question that you'd like to make before we adjourn this meeting?
I would say reach out to each other.
Sure, I
Really
will.
need to do this together.
Yeah,
So
yeah,
if you want, yeah, if you think you can give me advice, please do it.
yeah. I will send you an email, separate email, Rick, and after this meeting, I can easily look you up online.
Yeah,
I'll send you an
good.
email.
Yeah, I'm connecting to through LinkedIn already with you.
Yeah,
Yeah,
yeah.
yeah, so that helps. Yeah.
Shall we share the documents, Yina, and then you can spread it with the group? Share it with the group? How do you want to do that, the presentation?
Oh, the Rick, if the Auke, Rick, if you can, share your presentation if you don't mind. I don't have my note taker here today. Not that I have it. Usually I have
Oh,
my note taker, AI note
author,
taker,
yeah.
having this, but I think I forgot to, do it. But I'm recording this, so I'm gonna make
Yeah.
this and record it and the version available. I'll send it to Anarita so that she can upload it in the AgBioData YouTube channel, so that others can watch it. But I will send that presentation to Anarita as well so that she can share with the larger group.
Right.
In the meantime,
Yeah.
I will reach out to you, send you email week and Peter, so that we can schedule, follow meeting to discuss it
Oh,
for
yeah.
the collaboration.
One maybe last pressing question, Fabian. Are you working at Fraunhofer?
Yes, I'm
Okay.
working at Fraunhofer. Yes.
Stefan Gerd, you know him?
Yeah, he's my colleague. He's in the neighboring departments, so we are working together. Stefan is in the X-ray community,
Oh.
as you know, and we're doing the optical sensors and optical sensor analysis. So
Yeah, okay, just to keep it short, but Stefan is also involved in this how to implement MIAPPE challenge discussion group on with around 15 people from Australia and US and others. So we are linked.
Okay. So I will definitely talk to him about this so that we can make it.
Yeah, we got a meeting scheduled next week.
Okay.
Loop is closed. Good.
Okay, perfect.
Okay, thank you everyone. For those of you in the United States, happy Thanksgiving. And we will meet again in 2 weeks. And I will send you the reminder email a few days before.
Excellent.
Excellent.
Thank you,
you,
everyone.
Gina, and
thank you.
have a great Thanksgiving for those in the US.
Thank you for a great presentation and discussion on Auke week. Thank you.
Yeah,
yes.
welcome.
Thank you for the presentation.
Okay.
Yes.
Bye.
See you.
All right.
See
Thank
you.
you. See you. Bye.